Show my screen now.
I think you, as soon as I've introduced you, I think you can just share your screen.
Okay.
Okay. Okay,
yeah, for the main session first, Gunnar is gonna introduce you.
Okay, I'm going to start now. So hello everyone, welcome to Neuromatch 3, to the afternoon session. I've got 3 exciting speakers here with me today. I just want to make you aware that there's a question and answer tool. Please for the audience, please take advantage of these. We encourage a lot of questions for each of these speakers. And let me introduce the first speaker. First speaker is Aviv Emanuel from Tel Aviv University in Israel. And he's going to talk about inhibition of supplementary motor area effects distribution on effort over time.
Okay. Let me just show my screen. Okay. So thank you very much, Gunnar. And as Gunnar said, generally my topic is effort allocation over time, which means that I mainly tackle everyday issues, such as how do we maintain our focus during a long class or a long talk, how do we persist in working out over a period of, let's say, a few months or so. And mostly from everyday experience, if you go and ask people, they say it's pretty hard thing to do. People generally find it hard to persist in prolonged endeavors. And accordingly, also, effort allocation models suggest we say, okay, we have theories which suggest that there's a depletion of either a mental resource over time or a physiological resource, or maybe even accumulation of cognitive action costs over time, and even physiological substrates. If we're doing like a very tedious physical task, like lifting weights or going for a run. And we also have motivational accounts. For example, our motivation might shift from exploiting in general a certain resource or a certain task to exploring other tasks or other goals. And this happens over time. So our motivation to perform a certain focal task decreases. So generally, all of these models predict that effort decreases over time. We would exert most of our effort at the beginning of our task, and generally, we will gradually decrease our effort exertion. We can just test this prediction out simply by looking at world record performances in track athletes. So this group took world record performances from over 80 years and just observe how fast these athletes run. So on the x-axis, we have a division of intervals for each track. And on the y-axis, we have a running speed for each interval. And what we can see that from 1 mile on, people exert something rather, I think it is not predicted by the previous models. People initially decrease their effort exertion until the middle of a task, but then they increase their effort exertion again, near the end. So we get some sort of a U-shaped pattern, which is not predicted by most prominent models, both in psychology and psychology. This pattern of behavior is called the stuck in the middle effect, or U-shaped pattern in effort exertion, and exists in also other fields, athletic competitions, for example, in swimming, in cycling, and in rowing, and also in lab studies. Also, when we observe cognitive behaviors like correcting typos over a series of 9 articles, people correct typos at the fastest rate at the beginning and end of a task, but not in the middle. And also when people exert voluntary contractions, muscular contractions, when they asked to repeatedly perform these muscular contractions, they also exhibit this U-shaped pattern. So why does it happen? We have a general theoretical mechanism for this behavior, which is kind of elaboration of the most prominent models I discussed before. And without going into too much detail, I can say that it is generally claimed that the action value, I mean the value of performing a certain action, is at its highest near the starting point and the ending point of tasks. Meaning that closer to the starting point of a task, my action value will be perceived as very high. And during the middle of a task, I will perceive my action value as going downwards, and again, near the end of the task, action value will increase again. So the starting point and end point act as references for action value. Again, without going into too many details. Now, so far, for constructing a comprehensive model of effort exertion or effort allocation over time, we also need a neural mechanism. It would help if we would get like a neural grasp of what underlies this behavior. But so far we have a little bit problem with this issue because most of motivational neuroscience employ relatively short durations of behavior. It mostly concerns effort-related decision making and not effort exertion over prolonged periods. It mostly employed correlational methods, which is okay, which is very, very insightful. But we can't tell what causally causes certain behavior or certain effort allocation pattern over time. So we have to develop our own paradigm. We try to develop a paradigm for the study of these U-shaped patterns that enables us to study it in the lab. So participants had to play this game, this spaceship game. They had to control a spaceship appearing in the middle of a screen. They controlled the spaceship using the arrow keys, and they had to shoot using the spacebar. Each accurate shot in an asteroid gains them 10 points. And at the top right corner of the screen, you can see your score. And at the top corner of the screen, you can see a timer and the progress bar, indicating how much time you have left until the end of the clock. Now, your goal, or participants' goal, is to achieve as many points as possible, because we will generally get a monetary reward if we reach a certain criteria. The criterion was unknown to them, and was just we told them if they reached or not in the end of the experiment. So I jumped a little bit ahead. This is a typical result. This is the result of our pretest. We divided the time, the 10 minutes block, into 7 equal time segments. And on the y-axis, you can see the frequency of spacebar presses, which was standardized within participants. So what we generally see is this U-shaped pattern in key presses. The spacebar is the key that enables you to shoot asteroids. So this was our measure of effort exertion. And this pattern also replicates in other pretests, other experiments. We conducted 3 more control experiments, which indicated that this U-shaped pattern does not appear when the task lacks a clear endpoint, when the task lacks a timer or progress bar. And the U-shaped pattern does not appear when the task is too short, for example, 2 minutes or less. So we try to next just to assume what can be the neural underpinnings of this kind of pattern. And we thought the supplementary motor area might be involved or might be in play in certain aspects of this task because the SMA was thought to be involved in the coding of effort costs. For example, if people anticipate their effort to be higher, the bold signal from the SMA increases, and also TMS, inhibitory TMS to the SMA, was found to reduce participants' effort perception in a subsequent squeezing task. So our task, what we thought, is that if we have I wrote it down, but it's not important. We're not going into too many details. We thought in the present experiment that if we can inhibit the SMA, we might somehow alter the change in effort cost over time, which is reference dependent, and therefore cause a change in how effort is allocated over time. So what we did, we took participants to 3 sessions. On the first session, we had a task familiarization, which was a 10 minutes block and localizer scan, which they had to tap their fingers. On the next session, we had SMA stimulation, RTMS at 120% of resting motor threshold.
We have 2 more minutes.
Oh, no problem. It's perfect. Which is the inhibitory RTMS lasted 15 minutes and followed by the task. The inhibitory RTMS to the SMA. And up next, we had the control simulation to the precuneus, and which was followed by the task. This is what we got. Again, in the x-axis, we have a division of the 10 minutes block to 7 equal time segments. On the y-axis, we have standardized key presses. And what we found is that the SMA, the increase in effort in the SMA condition, which is represented by the black line, was steeper than in the control condition. And we also validated this result by comparing the SMA condition to the block from the familiarization session, in which no stimulation was administered. So to conclude this short talk, we can say that the SMA is involved in real-time action value coding rather than discrete decision-making, discrete cost of effort-related decision-making. It changes over time, and also inhibiting the SMA caused the value, assumingly, value of effort to change to a larger extent. Furthermore, we can say that the more the SMA is active, the more equally effort is distributed over time. So it might have some functional advantages. We might not want to fluctuate our performance over very long periods because it might be not so smart energetically, or just assumptions. And also we have the basis for further theoretical development of more comprehensive models of effort exertion. So I want to thank my advisors, Nira Lieberman here, and Nitzan Zenzo, Jasmine Ashage. Nitzan and Nira, I think these are pictures from them from 10 years ago, but Jasmine, she looked about the same. So if you want to reach me out, you can. And now if we have some time for 2 questions, I think you can.
Well, thanks so much. Thanks so much, Tel Aviv. Are there any questions from the audience? In the moment, I can't see any questions. Okay, if there are no questions, then I have one question, Aviv. Can these results be explained merely by motor inhibition?
Well, we ran a control analysis to tackle this issue, and actually, let's say if I can show. Okay, so actually, if we have accounts, motor inhibition accounts, would predict that the less the SMA is active, the less people are able to move their hand, and they should press the spacebar less. So we compared the rate of key presses across the whole block between the control and assembly conditions, and we found they were pretty much similar. Also ran a Bayesian t-test, and there's a small evidence in favor of a null effect. Also, spacebar pairs were highly correlated. So it kind of devalues effort inhibition effects, I hope. Any more questions?
Hi, Aviv. Can you hear me?
Yeah.
Hi, thanks for your talk. I thought it was really interesting. I've never seen a kind of U-shaped curve like that for efforts over time. Do you have like any intuition about why we've evolved like that? Why it's better to put effort in the beginning and end instead of the middle?
Well, my intuition is that we're pretty much programmed towards short-term benefits other than long-term. So if we have like a long-term goal, we don't plan accordingly, right? So we're mostly affected by proximity to these spatial cues like starting point, ending points, and this enables us maybe to maybe tackle multiple goals at once, just solving what's most important right now, the task with the shortest time frame is most urgent, and then we can achieve multiple goals. This is my general intuition, but I don't know. You need to run more experiments, maybe. I hope it answers this question.
Yeah, thank you.
Okay, well, there was another question from Gunnar. However, we have to stick to the schedule. So thanks very much, Aviv. And I'd like to now introduce Caroline. Caroline is currently a postdoctoral researcher at the University of Oxford and in the speech and brain lab. And today she's going to present part of her PhD in which she is interested in the function of M1 GABA and with respect to the changes in functional connectivity and retention in an adapted state.
Thank you so much for that introduction and also for the opportunity to share my work. So I'm Caroline Nettekoven. As Gunnar mentioned, I'm a researcher in Oxford and I'm interested mainly in the role of GABA in adaptation. I did my PhD with Charlie Stagg and Ned Jenkinson here in Oxford, and I'll be presenting part of my PhD work in which I link M1 GABA to retention of an adapted state. So motor adaptation is fundamental to navigating our world because our environment and our bodies constantly change, and to keep up performance, we're required to adapt to this change. So for example, since you've all had to recently work from home, you might have had to get used to using a different computer mouse. And this mouse might have different properties such as a lag, and you will start make movement errors in the beginning, like you can see here on the graph in the red curve. But you quickly adjust your movements to those new properties of your mouse, and your performance will return to former levels. Then once you've returned to your department mouse after some delay, you'll start making movement errors again, this time in the opposite direction. And this is because you retained some of that compensatory movement. So in my talk, I'll be referring to the phase where people change their movements in response to a perturbation as the acquisition of an adapted state. And then I'll be referring to the phase where the perturbation is removed after some time and people display an after effect that is gradually reduced as the retention of the adapted state. Now, there's a framework of adaptation that suggests that the acquisition of an adapted state adaptation task primarily involves the cerebellum, whereas the retention of this adapted movement relies on M1. And this is in contrast to other forms of motor learning, such as, for example, motor sequence learning, which is thought to primarily involve M1 at the acquisition stage. Now, in motor sequence learning, we know that GABA concentration measured from M1 early correlates with acquisition of a motor sequence. So we therefore hypothesize that in adaptation, where M1 is involved in retaining the adapted state, early M1 GABA concentration would correlate with retention. And then secondly, it's been shown that functional connectivity between left M1 and the right cerebellum changes after performing an adaptation task. So specifically, the activity in left M1 and right cerebellum becomes less synchronized. So here you can see pre and then post. So there's a decrease in the connectivity. So then we also wanted to test whether retention would correlate with the change in connectivity between left M1 and right cerebellum. So in order to test this, we had 15 healthy right-handed participants attend 2 MRI sessions where they were either scanned during a rotation task or during a control task. And I'll show you the rotation session first. So inside the scanner, participants controlled a cursor on the screen using this joystick with their right hand. They were instructed to hit one of 8 targets. So one of them you can see here. And this would be the optimal movement, and this would be the slightly imperfect movement that the participant needs. Now, after these baseline trials, we would impose a 10-degree rotation. So you can again see optimal movement, participant movement, and this time this is the visually rotated feedback. And we would increase that rotation every 40 trials by 10 degrees. So we end up with a 120-degree rotation at the end of the session. Then outside of the scanner, after some delay, we took the rotation away to assess retention of the adapted state. To look at M1 GABA, we collected magnetic resonance spectroscopy data from a voxel positioned over the left M1 hand area. And we also collected resting state fMRI data before and directly after performing the task. Now, the only difference to the control session was that in the control session, there was no rotation imposed at any point during the task. So here I'm showing you the MRS blocks acquired from the left M1 hand area, and each block consisted of 53 spectra that were averaged. And the block where we got our early measure of GABA from was this baseline block, so where people are not yet performing any kind of differential task between the sessions. Then I'm showing you the voxel placement here. So between sessions and between participants, across sessions and across participants, we got quite consistent placement of the voxel. And I'm also showing you the raw spectra that we're getting and the fit of the spectra. So we quantify GABA by fitting a linear combination of the spectral contributions that we expect to be there in the MRS voxel. And from this procedure, we obtain our GABA measure. Now I'm showing you the behavior here. So what you can see in blue is the control session. People are just fluctuating at around 0. And then in the rotation condition that you can see in red, when we impose the rotation, people make a 10-degree error, but they actually manage to reduce their error quite consistently over the course of the experiment. So from this phase, this main task phase, we got a measure of the adapted state by just calculating the average angular error. And then from here, we're getting, from the rotation condition, we're getting early during the washout period, we're getting a measure of the retention of the adapted state. Now, what we wanted to check first, because it's a slightly unusual adaptation paradigm, was to see whether we're actually driving changes in the brain that are known to occur in response to adaptation. So specifically, we set out to replicate this paper by Albert and colleagues, where they show that after adaptation, but not after control, the functional connectivity of a cerebellar network is increased. So in our experiment, that's exactly what we also find. So I'm showing you here in magenta the cerebellar network just as a mask. And what you can see in red and yellow are voxels that are increased after performing the adaptation task compared to after performing the control task. To also test the specificity of this result, we ran the same analysis on the default mode network where we didn't expect any kind of changes, and indeed we didn't find any changes. What we also did was then extend this analysis that Albert and colleagues did by looking inside the rotation condition and seeing whether we can find a relationship between the strength change of that network and the actual adaptation performance. And that's what we're finding. So what you can see here is people having a lower error, so performing better on the adaptation task, they also increase the strength of the cerebellar network. And we don't find any kind of similar relationship for change in strength of our control network, and these are significantly different. We also looked at whether our results hold when we quantify adaptation using a commonly used state space model, so model-based approach to quantify adaptation performance. So we modeled the adaptation performance on this task using a single state model. And we find that our model-free and model-based parameters of adaptation correlate highly, and that our results hold for modeled adaptation as opposed to just an angular average error. So that gave us some confidence that we're actually driving changes in the brain that are known to occur in response to adaptation, and that our measures of adaptation, it doesn't matter how we quantify adaptation, the same results hold. So next, we actually tested our specific hypothesis regarding early M1 GABA and retention of the adaptive movement. So what we find is that M1 GABA measured at baseline correlates with retention, but not with adaptation. So the higher your M1 GABA concentration at baseline, the more you retain of the adapted state later after adaptation. We also see that the change in connectivity between left M1 hand area and the right cerebellum, this change in connectivity also correlates with retention, such that if you decrease, the more you decrease your connectivity, the more you retain of that adapted state. And then lastly, what we see is that there's a relationship between M1 GABA and this change in M1 cerebellar connectivity. So kind of having a threefold relationship between the three measures. And this relationship does not exist between the neurotransmitter and change in connectivity for a control neurotransmitter, which is glutamate in this case. So because we got this threefold relationship, we wanted to further explore this, this data, and therefore calculated a mediation analysis. So a mediation analysis tests whether the observed direct relationship between two variables, so in our case, M1 GABA and retention, might be mediated through an indirect pathway. So in our case, M1 cerebellar connectivity change. And that's exactly what we find. So our mediation analysis showed that the relationship between M1 GABA and retention was mediated by this indirect path, so connectivity change between left M1 and right cerebellum. So to go back to our original question of what is the role of M1 GABA in retention, this is the kind of model that emerges. So higher M1 GABA concentration before adapting relates to a decrease in M1 cerebellar connectivity, which in turn relates to increased subsequent retention. So just to summarize all of this, I showed you that our adaptation task, adaptation increases connectivity in a cerebellar network in a way that is performance relevant. We find that there's a relationship between early M1 GABA and retention of an adapted state. And we also find that this relationship is mediated by the connectivity change between left M1 and right cerebellum. So with this, I'd like to thank my supervisors, Charlie Stark and Ned Jenkinson, for all their support, my lab group for their help, and of course, thank you to everyone who is listening. Thanks.
Well, thank you so much, Caroline. That was fantastic. Also, thanks very much for sticking to the timetable. We have time for a couple of questions. Are there any questions? Please feel free to get in touch, use the question tool here. Okay, there's one question here, which thanks, Caroline. Have you guys also looked at networks between the cerebellum and the posterior parietal cortex from Takeshwar Singh?
That's a very good question. I think it's an interesting question to address. We haven't looked at this yet, but that is something that we could do with our functional connectivity data. And certainly, I think the posterior parietal cortex would be an area that is likely involved here. But no, we haven't looked at this yet.
Are there any further questions from the audience? Yes, there is another question here from Nick Kitchen. It says, thanks, Caroline. Great talk. Is there any reason why you chose a stepwise permutation paradigm instead of a consistent rotation?
Yeah, that's a good question. Because I think the reason that we went for this kind of paradigm was really because we wanted to drive adaptation throughout the whole period of the experiment. Because as you can imagine, we were also trying to find out whether there might be any kind of change in GABA during this adaptation period, which we didn't see. And in order to see this kind of change in M1 GABA, you have to really give GABA basically time to catch up. So in a sense, drive the process throughout the whole time. So we really wanted to dissociate retention and adaptation here, and therefore wanted to be sure that we're driving adaptation for long enough to these changes to actually emerge.
Fantastic, thank you. There are a couple of more questions, however, we have to stick to the schedule. It's now nearly 1/2 past, so I would encourage the people in the audience who have questions to probably direct them directly to Caroline, and it's very easy to find her online. I did this morning. Now let's come to the third and last talk for this session, which will be given by Gunnar Blom from Queen's University in Kingston. He is also known as the Viking, and therefore he renamed or named his lab the Viking Lab, which stands for Vision and Kinematics Group. And he is also one of the co-founders of the NeuroMatch Academy, and brewing beer is one of his favorite hobbies. Please go ahead.
Thank you, Gunnar. Very happy to be here. So, to plan and execute reaches, the brain essentially needs to convert sensory inputs about the hand and the target into appropriate motor commands for the arm, including using information about which arm to actually use for the reach. So previously, we have combined magnetoencephalography, or MEG, with a pro-anti-reaching task to address this. And so participants essentially had to point towards a target, either in pro trials, where they had to point towards the target, or in anti-trials, where they had to point to the mirror opposite of the target, and that depended on the color of the target, essentially. And we used the brain's lateralization with respect to the visual hemifield on the body to ask which brain areas show differential activation that correlates with the sensory motor transformation, so the transforming essentially the target location into the appropriate movement. And we essentially determined if areas are correlated with sensory or motor information through differences between conditions. If only sensory information was present, then we expect the same activation for leftward targets regardless of the movement, and the same activation for rightward targets regardless of the movement. In contrast, for movement information, the same activation is expected, for example, for a pro-leftward trial and an anti-rightward trial, because those would result in the same movement. So when you subtract pro from anti and left from right, then you would essentially get a signal that is driven by sensory information. And if you subtract pro-left, anti-right from anti-left, pro-right, you would get this, the same thing for movement. So using this reasoning, we showed that right after target presentation, there's a feed-forward sweep of sensory information from occipital to frontal areas. And then motor coding essentially, or correlation with movement, showed up first in the frontal areas. And then we found essentially that afterwards there was a backwards sweep of motor coding from the frontal areas towards the parietal areas. And on top of that, this motor plan was stored in working memory as evidenced by sustained parietal gamma band activation during the delay period, but I'm not showing this here. So now remember that to produce a movement, we also need to know what effector to use. So this data is only using the right hand. However, little is known about the cortical network underlying the integration of information about which arm to use, and that's exactly what we're going to address here. So to address this, we use the data from the same pro-anti task as I showed you before, but now we record it from both hands in a block design. So again, either you had the left hand during a whole block of trials in a pro-anti trial, or the right hand. And again, this was a memorized reach with about 1.5 seconds between the onset of the cue, so the spatial cue, and the onset of the movement cue. And so we then looked at the difference in oscillatory power between the pre-target baseline and any time point after the target onset. And so this is what's shown here. And since we're interested in the integration of hand information with a movement plan, I will only show movement aligned results here. But we also did, of course, a sensory aligned analysis, but there was no significant effects there. So here's an example of the desynchronization that you would get essentially in the beta band power here, leading up to the movement. And so you can see that essentially prior to the movement onset, which is at 0 here, you get this desynchronization in left primary motor cortex when the right hand is used. And this is for a pro-leftward target. Now, if we look at the same thing for a rightward target, you see pretty much the same thing. And that is simply because we cannot, or MEG cannot resolve left versus right movements in this case. So we can essentially average across these two, for example. Now, you can do the same analysis for the data of the left hand. So if you do that, then you can see, so this is again in the left primary motor cortex. And since the left primary motor cortex mostly codes for right hand usage, you can see there's much less of a desynchronization here. Okay, so we will now perform essentially a subtraction analysis to compute a single summary result for hand use coding in a given bilateral brain area. And so then we can essentially average across different conditions here. So the way we do that is that we essentially have the same as on the previous slide. So we have the left M1 right hand use, the left M1 left hand use, and then we can subtract them to get the left minus right hand for the left M1. We can do the exact same thing now for the right M1, and you can see that there's the opposite result here from the left M1. That's simply because the subtractions is done in the same way. And then if we subtract essentially left M1 from the right M1, we get a single index essentially for bilateral primary motor cortex of what's going on. And we can then time resolve that a little bit differently here by just looking at the 20 hertz of beta band activation and asking essentially when that becomes significant. And you can see that very early on, prior to movement onset, there is evidence for hand information being coded in primary motor cortex. Okay, so next we want to essentially look at the whole brain map of where this happens. And this is shown 500 milliseconds prior to the movement. So here's what we see. We find essentially that hand use related beta band activity changes in frontal parietal areas. And we also find alpha band changes in more posterior areas. And note that alpha band is typically associated with sensory processing. Okay, so next we want to know how the hand information was integrated with the motor plan. So we computed essentially the motor information separately for left and right hand. And we argued that if there was a difference between both, then this is indicative of a hand-specific motor plan. What I'm showing here is the difference for left M1 between the left and the right motor plans. And so you can see that there is some differences again in the beta band showing up. And if you do the same thing for the right motor cortex, the opposite happens here, which is essentially to say that the same thing happens, but due to the calculation, the sign is inverted. And then we can again essentially compute by subtracting left and right M1 an overall index of the interaction between hand and movement effect. And that shows some significance before the movement onset. So looking at the whole brain map in the 500 milliseconds prior to the movement again, we find that we have hand-specific motor plans in a more sparse network than hand representation of the brain. And this is only the case in the beta band and not in the alpha band. Now, we can also look essentially at, or to summarize those results, we can highlight the areas that show movement information in pink. So this is from our previous study. And now overlay essentially the hand specificity in blue and whether motor plans were different for each hand in green. And so you can see that different subnetworks are involved to represent hand information and hand-specific motor plans. And then we also looked at the timing of these signals. That's the nice thing we can do with MEG. So here is essentially the detection of significant hand information and hand movement interactions. And you can see that different brain areas essentially show this at different points in time. And there's no order if you compare, for example, the hand coding and the hand motor interaction here. And then if you overlay that to our previous studies results with respect to when a motor plan became apparent, you get essentially a pattern like this. And from there, we can essentially summarize by saying that we found independent hand and motor plans in the parietal occipital junction and dorsal premotor cortex. We found hand use only, so no motor plan in the superior parietal lobe and primary somatosensory cortex. And we found a hand-specific motor plan in the superior parietal occipital cortex, the interparietal lobe, the ventral interparietal sulcus, and primary motor cortex. And so maybe I can also say that given that there was sort of like different aspects involved in motor planning, we found essentially different subnetworks that were involved in those different aspects of the sensory motor transformation and movement planning. And maybe the reason why we found so many brain areas typically activating when we ask participants to do a simple reaching task is that we don't probe all these different aspects, like which hand to use, posture, decision-making, rule coding, et cetera, et cetera. And so this would be our hypothesis to say, or our prediction to say, is that if you were to tease those apart, you would find different overlapping subnetworks that all contribute to reach planning. Thank you very much for your attention.
Well, thank you very much, Gunnar. Thanks very much for the interesting talk. For those who are interested, I've also put the associated paper into the chat where you can look this up. Okay, then there is a question here, but Caroline, I suppose that is related to one of your previous interactions with the audience, or could you unmute yourself, Caroline?
Which one are you referring to? Which question?
The one that is just appearing in the chat.
Sorry, that was yeah, I was answering JJ.
Okay. Then there is no problem. Then there's a question here from Bernard Marius Hart. Is there an effect on practice on the activity patterns? Gunnar.
I have not looked at that. We see, so if you look at, the experiment was a blocked design and we used different postures. And if you look essentially at different postures, early versus late in a block, there's no difference. So I have split the data, like just like the first, there was always 400 trials in a block. So the first 200 versus the last 200, I essentially find the same effects. Although I must say that I might not have the statistical power to resolve any more subtle differences there.
Okay, thank you very much. I hope that answers your question. Are there any more questions with respect to Gunnar's talk? We have still time for questions. Okay, then how could different arm postures, how could they maybe change the motor planning, Gunnar?
Yeah, so that's a very interesting question. It's actually something we have the data for, but I haven't analyzed that yet into too much detail. A little bit though. So we can, the idea is essentially that if you do these pointing movements like this, right? You can also do them like this. But if you now flip your arm around, then this movement here, which now goes to the left, essentially would go to the right in this posture, right? So essentially, there is a posture dependence on the spatial effects of your muscle activation, and that should be reflected somewhere in the sensory motor transformation for the movement planning. And we're still analyzing that, and hopefully maybe next year at Neuromatch conference, I don't know what that will be, 6.0 or something like that, I will be able to give you the results.
Fantastic. Well, thank you so much. Well, then if there are no further questions from the audience, from the attendees, then I would like to thank all the speakers for their interesting talks. And yeah, I wish you all a very good day. Okay.
Thank you, Gunnar.
Bye.
Thank you, everyone else. Thank you, Taha.
Bye.
Hi, Michael.
Hi. How are you?
I'm doing good. Hold on. Okay, I see your video.
Hey, great. Fantastic.
For the other two speakers and the MC to join in.
Okay.
Done some tests before we start then.
Sure. Yeah, for sure. I'll just stay here. So.
Okay, Aina has joined. She's MC.
Okay.
Can you just share your screen once to see everything's all right?
Sure. Yeah, I'll share my screen.
Hello, Aditya, and hello, Michael.
Hello. How are you?
I'm very good.
So,
And
this is also being broadcast live on YouTube right now.
Okay. So can you both see my screen? Everything's
yeah, it looks very good. And just see it. Now you're in screen mode. Yep, that's looking good. Do you have any videos or any audio that you want to test?
Nope. No, I think I'm good. Thanks. Yeah. That's
Oh,
it.
very good. And I like your background.
Oh, thank you. Yeah. Figured vary it up from the space thing I've been seeing all over here. So
yes, I'm making my own backgrounds too. And this one, of course, I've got the company brand behind me now. So otherwise I would have other backgrounds too.
Yeah, very nice. Yeah.
Yeah, they did a nice job. That was Gunnar Blom designed that one. And he's got a number of variants, but this was the one that people wanted us to use. So that's good. So yeah, so we were doing a little bit of homework on all of you, and I found some of your paper, even though it's not related to what you're talking about today, your recent paper, but we're going to give it a plug anyway.
Oh, thank you. That's so kind.
We're going to do that with everybody. Everyone's got a recent paper. It's really interesting and exciting, so we thought we'd do that.
Oh, thank you. That's so nice. Yeah, it's not quite related. It's still sort of the intersection of computational stuff and synaptic.
Right.
Stuff, but
okay. I better put my phones in so that we don't have an echo.
All right.
Just to confirm, what's the title of the paper that you're talking about now?
Oh yeah, I put those in the chat window in Slack for you, Aditya.
I have 3 of those. I just wanted to confirm which one right now.
Okay, though, let me look in Slack and I'll tell you which one it is. Oh, actually, let me look in. I've got them up here. The one that we want is the very first one. Well, Michael's paper. Whoopsie. Michael's paper that I found is the one in Cell Reports, a synthetic likelihood solution to the silent synapse estimation problem.
Okay.
So that's Michael's one. And then we've got Chris Wu's paper, and that's the PNAS one, the homeostatic mechanisms. So that's the first speaker.
Okay. And there's another Zoom Room 7 host here.
Hello. My name's Taha. I will be the host for this session on the back end. Dr. Fuse, you are the main host. You are the MC.
This is the second time this has happened today. I was actually booted out of an MC position because there were 2 MCs, and now Aditya. Aditya had originally emailed me, so there's a little bit of confusion here, and honestly, it's better than 2 hosts are better than 1, and 2 MCs are better than 1. But I'm not sure which one is the right one, because
I'm not the host. I'm not doing anything, Dr. Fuse. I'm just trying to keep this tech side going on, and for the green room, as it is called now, before the webinar starts. I will just make sure I check in with everybody, all the attendees, and make sure everybody's mic works out. they can ask Q&A, stuff like that. So before we actually run out of time, why don't we do this? The ground rules, Dr. Puce, you might have, laid them out already in the past few seconds before I joined in. But I will just add some more stuff to that. Is, as you know, this is getting streamed live to
Hi, Vicky. Vicky,
YouTube.
you just joined us at the right time. Taha is giving us instructions, so you may want to listen to what he's telling us about, okay? And you can hear us great.
Yeah, just a few technical ones. Okay, perfect. So everybody here will on their turns go with the presentation. The timeline is absolutely strict, as has been discussed before. Just wanted to say it is streamed live on YouTube. I will provide the link as soon as the webinar starts. So that, if somebody has connectivity problems out of the attendees, they can just switch to that one. Secondly, for the presentations, we will go with the share screen type. For the Q&As, I request that as soon as your talk ends, you can stop share. And for the Q&A, we can all go to a gallery view and have a chat with the attendees. If you need to go back to a slide, you can share your slide again. And if you would like to do local recordings for your purposes, absolutely fine. Feel free to do that as well. And the other thing that I would like to add is, for everybody, if it's possible, we can go on the arbitrary order that's on my screen. Everybody can share the screen and unmute for a second, and then just leave it.
All right, so let me share my screen first. Recursiveness there. So let me unshare and mute.
Perfect.
Yeah,
Perfect.
okay, I'll do it next.
Yeah, because you're first.
Yeah, okay, share my screen. And
it's good. And then Michael is next. Michael's presenting second.
Great.
And we've tested Michael's already. Yep.
That's true.
And you can mute as well, Michael. Good. And then Vicky, why don't you see if you can share your screen?
I just wanted to add something before Vicky shares as well. I think I'm in the wrong room.
I think you might
from
be.
Zoom room 9. Okay.
You better go.
I will just get out.
But thank you.
Okay, but we have the actual host down there.
I'll find a room that you're in and bomb
Yes, Aditya. Yes. Apologies. I just got the message.
This is great. I love this. Okay, so Vicky, can you share your screen, please? It's okay, Aditya, because I knew that you were the person who originally was supposed to do this. So great.
Yeah,
and
I think, did everyone see it?
Everyone can see it. You'll just have to go into presentation mode and then see if you can mute yourself.
Oh, okay.
Yeah.
Wait.
Just hit the mute button and just see if you can mute yourself, just in case there's any background noise anywhere.
Okay, so then if I mute myself, how am I present?
Not now. I mean, when,
not now. No, we're just testing. We're just testing to see if everything works.
I see. I see what you're saying.
Yeah. Vicky,
okay.
Can you press play and see what you see on your screen? We just want to ensure that we don't see the presenter view. Like, we want to see the slides and not the.
Yeah.
Oh, like this.
Yeah. Please, yeah. Just so if you hit play on your screen share, because you shared your screen, and then if you hit play, then it goes into presentation mode in your keynote. Beautiful.
Yeah. This is great.
Beautiful. Lovely. Okay, so here are the ground rules, folks. We have 15 minutes max for each person. After 10 minutes, I might I'll just interrupt and say that's 10 minutes. And then after 12 minutes, I'll say that's 12 minutes. And then when 15 minutes comes, I'm muting you. because that's what we've been told to do. So I'm sorry, but that's what we have to do. So,
so are you going to say, like, actually physically say 10 minutes or 15 minutes like that?
If you would like me to, would that be helpful to you?
One thing that can be done is that Vicky, all the speakers, when you share your screen, you'll be able to see Aina's video on your screen.
I will be able to.
Yeah, it'll be down in the bottom left corner for you. So what I'll do, I can go like this if you want.
Okay.
Would that work?
I don't think I will go over, first of all.
Well, but we have to have a plan.
Yeah, sure.
We have 3 minutes.
Okay, I'm going to go like this for 10 minutes. And then if there's for 12, I'm going to go like this. And then I'm going to go like this for 15. And then I'm going to mute you. And what we'll try and do is we'll try and ask questions. We're going to be watching the chat, the question Q&A thing. We'll see what's upvoted. And so we don't have enough time to allow people to come in on the stage to do it. I'd prefer to do it, but we don't have time. So I'll ask the question on behalf of the person for you. We probably only have time for 1 or 2 questions each one, okay? Mm-hmm. And then I'm getting now a practice session. Attendees cannot join until you start the webinar.
Yeah.
Aditya, when do you want to do that? We were told to start it a couple of minutes early.
Yeah, I just want to get one more thing out. If anything you want me to share during your talk or before your talk on chat with all the attendees, you can just put it as a message on the chat bot here right now.
Yeah, and remember on your chat, folks, you can either message Aditya, the host, or me, or the other panelists, or the whole audience. So just be careful which one you message. But Aditya, I've given Aditya your most recent papers, all of you, which we're going to advertise in the chat. And Vicky, your paper, recent one, is relevant to what you're talking about today. For you, Chris, and for Michael, no, but we're going to just put it up there anyway. All right?
Thanks.
Okay.
Thank you. Yeah.
Awesome. I'm going to go live now.
Okay, this is it.
Okay. Yeah,
all right, I'm ready.
Okay. Should I share my screen now?
Not yet. We're a couple of minutes early, and what we're doing is we are streaming live right now, and we are waiting for the live stream to come in. There's a little bit of a delay, so we start a little bit early, and then I will just give you, Chris, an introduction, and then we'll be able to launch into it. But it's 13:59 right now, and people are starting to probably join the session, and just slowly, and we'll see how we do. We'll try and get out of here on time. I'm looking forward to this session because there's some really interesting stuff in this session. In fact, I was doing my homework on the presenters in this session earlier, and in fact, it's gone 2 o'clock, or at least 1400 my time here. So we should get started. Let me just do a brief introduction. My name's Aina Puce. I'm going to be the MC today, and we are in the very capable hands of Aditya Iye, and he's in Zoom room 7. So he's in charge of all of the back-end stuff and making sure everything works for us. And we have 3 very exciting presenters on the neuroscience side, and it's a pleasure for me to run this session. Our first person today who's going to speak to us is Chris Wu. He is working on some of the idiosyncrasies of the olfactory system, and I'm actually very excited about hearing about this, because the olfactory system is the Cinderella of all of our sensory systems, and we don't know enough about it. In the human brain, at least, which is what I study, we don't understand the mapping of odorants to cortical regions. And so it's really good to see that people are working on this kind of problem already. And so he's going to tell us about olfaction. And so my take-home message for you is olfaction is the new black. So without further ado, you, Chris Wu, I'd like to invite you to start your presentation. Thank you.
Okay, thank you for a nice introduction. Okay, so today I will talk about super neural network with a short-term plasticity can account for transient neuronal dynamics in the olfactory system. The olfactory system is widely considered as auto-associative memory networks, which can generate similar representations in response to chemically distinct but functionally related odorants. For instance, lemon and lamb smell similar. So classical auto-associative models rely on strong recurrent exactory connections accompanied by attractor dynamics. So this is a simple network energy landscape. In this case, there are two attractor states, and the input drives the network into one of the two states. Since this is the fixed point of the system, the network will stay in these attractor states, therefore exhibit persistent activity even after the stimulus is removed. However, the persistent activity is not observed in the olfactory system. Instead, the new response in the olfactory system is transient. So here's one study by Kevin Balding and Kevin Franks. The optic gyrate stimulates the olfactory bulb, and they use electrodes to record the neural activity in the piriform cortex. Those are the responses in the piriform cortex to different light intensities. You can clearly see that the response are transient with huge peaks. And this experiment is done in mice, but the transient response is also observed in other species. For example, here's another study by Peter Hubrich and Rainer Friedrich. They did this experiment in a zebrafish. So here they present the odor for 10 seconds, but it's only evoked the transient EPSCs and the IPSCs. And furthermore, a lot of experimental evidence indicate that the network is operating in an inhibition stabilized regime during stimulation. So here we are interested in what is the underlying mechanism of transient neuronal dynamics. So to address this question, we use a superlinear network framework in which the neural input-output function is a superlinear power law function because it's analytically tractable, and also this superlinear function fits the intracellular recording data quite well. So let's first consider about a simple scenario in which the network consists of one exactory population and one inhibitor population. We can write the network dynamics by following in equation. Those are quite standard equations. So alpha E, alpha I, those are the powers of the input-output function without a loss of generality, and we'll keep alpha E, alpha I as they are. For the results, I will show alpha E, alpha I are equal to 2. So here we are here we are focusing on network with strong recurrent exactory connections. In other words, this JE is large, such that the determinant of the width matrix is negative. And we are particularly interested in how the network responds with the input GE, because this mimics the older stimulation what I showed in the experiments. So how does the superlinear network respond to input? So here is the results. X-axis is the time, Y-axis is the Fahrenheit, and exactory in blue and the inhibitory activity in red. So during this green bar, we add additional extra input to the exactory group. As here you can see the network activity explodes. So we can also plot the physical track before stimulation and during stimulation. So before stimulation, there were two fixed points, one stable, one unstable, and the network was initially in this stable fixed point. And but if we increase the input, the fixed point disappeared. So if you think about this activation of exactly population as a kind of memory pattern, then it's functionally good because that means the input can successfully trigger the activation of the input pattern. However, this activity is very high and the dynamics is unstable, which is inconsistent with the charging response what I showed previously. So this arises a question, which mechanism stable as the network dynamics, therefore generate this charging response. So we knocked it into three different mechanisms which operate on under a short time scale. So the first one is the spike frequency adaptation. The second one is E to E short-term depression. Last one is E to I short-term facilitation. So before I show you results, let's look at the stability condition of superlinear networks. So there are two conditions. The first one is the determinant of the weight matrix have to be positive. The second one is this condition. This relates to the sum of the eigenvalues of the Jacobian matrix of the system. So for E to E short-term depression, we have a kind of prefactor which multiplies with JE. So we can calculate the steady states and plug them in. As here you can see, if the activity, if the network dynamics is unstable, RE is extremely high, this is the first term will decay to zero. Therefore, the first condition is satisfied. A second condition is satisfied in the same way because again, this one goes to zero, and the rest is returns they are positive. So how about E to I short-term facilitation? For E to I short-term facilitation, we have another factor which is multiplied with the JIE. So similar as before, but here this value increases. So if this one increases, the first condition can be satisfied. A second condition is less obvious because it doesn't include any term, any JIE term. So, but we can rewrite the condition a little bit by dividing this term. And if we replace the fine rate by the current, we get an equation like this. And since we have alpha which is larger than 1, which means the numerator grows faster than the numerator. Therefore, this term will eventually decay. Therefore, the second condition is satisfied. So, as I mentioned before, we started with a weight matrix which is negative. Since the spike frequency adaptation won't change the syllabtic strength, therefore it cannot stabilize the network dynamics. Both E to I short-term depression and the E to I short-term facilitation, they can stabilize network dynamics. But the way how they stabilize are mechanistically different in terms of how they satisfy the second condition. So for E to E short-term depression, it doesn't need inhibition. But for E to I short-term facilitation, essentially how it does is actually increase the E to I synaptic connections, therefore increase the inhibitor fine rate, regulate the EI, therefore stabilize the network. So this is indeed the case, which is confirmed by the simulation results here. The spike frequency adaptation cannot stabilize the network dynamics, and the E to E can, but if we remove the inhibition in this green period, it can restabilize the network dynamics. In contrast, E to I short-term facilitation can stabilize the network dynamics, but if we remove the inhibition in this period, the network dynamics is unstable. So suggested by the experimental studies that inhibition is crucial, therefore we think E to I short-term facilitation is more likely to be the stabilization mechanism in this process. And in this process, when the network dynamics switch from stable to unstable and eventually restabilized by the plasticity, short-term plasticity mechanism, this generates a huge trending response, which is similar as we observed experimentally. So this is just the example of two populations. Of course, our brain store multiple memory patterns. We therefore extend this superlinear network framework to a network with multiple populations. So here, I'm showing you a toy example. So there are three populations, two executive groups, one inhibitor groups. And for network with multiple population, what we are interested in is what is the condition of having one single stable fixed point? Because for networks with multiple population, you can have stable network dynamics, but it's multi-stable. In other words, if you present a pattern but you withdraw the pattern, the network doesn't go back to the initial baseline, which is what we don't want. So here is the condition of having a single stable fixed point, which we call it UD stability index. If this one is positive, then the network is multi-stable. If this one is negative, then it's UD stable. We also define a kind of RS index, which is the leading eigenvalue of the E to E submatrix. If this one is positive, then the network is in inhibition stable as regime ISN. If it's negative, then it's non-ISN. So, so here I'm showing you one example corresponding to the negative UD stability index. As here you can see, here we present an input, the activity increase, but if we withdraw it, the network go back to the exactly its original baseline. And the ISN index also change. It's a switch from a non-ISN regime to an ISN regime. And for the positive UD stability index, the network activity won't go back to the initial baseline. So with this, I want to sum up. So I show that trending response can arise when the network dynamics switch from stable to unstable, and they eventually restabilize through stability mechanisms. And unlike spike frequency adaptation and the E2E shortened depression, E2I shortened facilitation is more likely to be the stability mechanism in this process. And the superlinear framework can be applied more generally, can be extended to multiple population. So yeah, thank you for your attention.
Thank you, Chris Wu. And as we wait for questions to come in, I wanted to just ask you, this is really fascinating work because there are three different mechanisms, yet the transient response is very, very stereotypical. And I find that really fascinating. Do you want to talk a little bit about that?
Yeah, so the trending response here we think is the network dynamics switch from stable to unstable. So in a linear network, this can be not achieved because the network stability is independent from the input. Here we study a case with a superlinear network. Actually, the network stability can be changed by the input.
Mm-hmm. Well, this is going to be a very really nice way to go in the future to test out and help people plan doing real experiments. And I think I can see the value in that, and that's excellent. So thank you very much for answering that question. We do have time for some additional questions. Please don't be shy. People are being shy today. And we have a little bit of time, but if people are going to be shy, then we will just move on to the next talk. So thank you, Chris. That was really fascinating and very thought-provoking, and it's made me excited to think about what else is going to come out in the field. Okay, so we switch from olfaction now, and we're still going to be talking about nonlinear behaviors. That seems to be the the main theme of this session. And that actually makes me very happy that people are actually featuring nonlinear behaviors. And the reason for me why it makes me happy is that I get concerned because so many of the analyses, the methods that we use in neuroscience rely on or assume linear behavior in the system. But why should there be? Now, we assume linearity because it's easier to calculate, but does that mean that that's the right thing to do? And that's a question we can discuss a bit later if we wish to. But Michael Lin is going to tell us about these dynamic filtering mechanisms in a inhibitory serotonin network and their nonlinear behavior. So Michael, the floor is yours.
Great. Yeah, thank you very much. Yeah, I'm excited to be here and present my my work. So go. So yeah, I'm going to be talking to you about an inhibitory serotonin network. And so this is work done during my PhD. Dr. Jean-Claude Bake is my supervisor, and this work was also done in close collaboration with Richard No, also at University of Ottawa. So yeah. So as a lab, we're really interested in dorsal raphe nucleus. This is a really fascinating midbrain structure. It's involved in affective processing. And it's interesting because it's a network hub. So it both receives inputs from large portion of the brain, and it also sends outputs to serotonin outputs to a lot of the brain. And so these are just some of the structures it communicates with. And obviously it's the target of antidepressants like SSRIs. So it's really important to understand how it processes input. So another structure I'll be talking to you about today is lateral habenula. So it's shown here, it's right below hippocampus. And habenula really represents negative valence signals with a really acute rate code. So it's really nice like that. So this is some monkey work showing that an air puff can really acutely increase firing rate, and these are graded responses. So depending on the strength of negative valence signal you give, there's really a graded response in habenula. This is just some more work showing that these neurons can burst very strongly. So we're really interested in dissecting this whole pathway and figuring out its involvement in mood processing and depression. And so just a brief overview, I'm going to talk about some synaptic physiology of this pathway. I'm going to be talking about dynamics of these recurrent serotonin connections, and I'm going to tell you all about that. I'm going to talk about information transfer through this pathway, and then finally some simple behavior and how we can use the predictions we've made with our network modeling to actually inform the behavioral results. So it's kind of a bird's eye view of things, yeah. So first, what does this input do? So we're just going to use some simple synaptic physiology first to characterize this. So we've injected habenula with an AAV expressing channel rhodopsin. We then make midbrain slices, we patch genetically identified serotonin neurons, and then we activate these habenula axons with short pulses of light. And so when we do this, we see something really classic. We just see a classic excitatory inward current. So this is AMPA mediated, can be blocked with NBQX. We show they're monosynaptic. We also show there's NMDA components. So so far it seems like really classic, monosynaptic excitatory transmission from habenula to raphe. But there's actually something really interesting we started noticing, which is when we activate the same pathway at high frequencies, we see something completely different. So again, this is low frequencies. You have this excitation, but high frequencies. This is a separate neuron, actually. So what you see is at high frequency stimulation, you see an outward current. So this is an inhibitory current. And if you look at the time scale, it acts over almost a second. So really odd. We did some characterization. We did some IV curves. The long story short is we showed this inhibitory current is actually mediated by 5-HT1A receptors. So that's kind of odd. So this is just showing that an antagonist to 1A receptors, Way100-635, can completely block this current. So this is kind of odd because 5-HT1A receptors are classically thought to be autoreceptors. So that means that it's thought that serotonin neurons essentially regulate their own firing rate by secreting serotonin in the extracellular space. It binds to these autoreceptors, and it effectively provides gain modulation of their firing rate. So that's the possibility up top. We actually show there's a recurrent inhibitory structure happening in this network. So that's the bottom. So these neurons actually inhibit one another. So we have sort of a lot of convergent anatomical and other evidence for this, but I'm just going to show you sort of one piece of that story that's probably the strongest evidence, which is if you inject an opsin directly in RAFA, and you inject it sparsely such that you have a sort of a salt and pepper mix of expressing serotonin neurons and non-expressing serotonin neurons, then if you record from one of the neurons that expresses this opsin and then activate the opsin, then you see nice spiking behavior shown here. But if you patch one of the non-expressing neurons, if the circuit is organized as a recurrent inhibitory circuit, then logically you should be able to patch a red neuron, a non-opsin expressing neuron, and still they should receive recurrent inhibition from the green cells. And that's effectively exactly what we see. So if you patch a red cell, you see this identical outward current, and you have no light-induced artifacts. So these neurons are completely non-expressing. So this is pretty strong evidence that there is actually a recurrent inhibitory circuit. We actually also completely rule out autoreceptor activation. So there's actually no component of the response that's due to autoreceptors. So next is kind of my favorite segment. So now we're going to be talking about information transfer through this circuit. So this is really cool. We found some really nice rules for this. So first of all, facilitation dynamics. So I just told you that high frequency activation produces this inhibitory response, but low frequency doesn't. So let's sort of dissect that a little more. So here we're still looking at RAFA, but we're electrically stimulating serotonin axons while patching a serotonin neuron. We have on board a cocktail of drugs to block everything else. And here we show that we can elicit an outward current, still blocked with the same drug. So it's still the exact same thing we were studying before. And we can do these frequency experiments where we keep the pulse number fixed, so still 8 pulses always, but we vary the frequency. And what we see here is pretty striking. So at 1 hertz, you see absolutely no response at all in these neurons. And as you increase the frequency, all of a sudden you start getting this really massive inhibitory response. So it's a really steeply frequency-dependent response. Here's just some group data. This is kind of a pain to characterize, as synaptic physiologists will know. You can't take the amplitude, so you have to integrate over current to get charge, and then compare that to a surrogate noise distribution. But you can get some nice results out of that. So from this, we really want to figure out what the role of this is for network processing. So we made a model of these short-term plasticity dynamics. So this is actually taking a model developed by Julian Rosbroich, who was actually presenting at the same time slot in another session, unfortunately, and Richard No. And so it's a linear and nonlinear model of dynamics. So effectively, there's a presynaptic spike train. There's some kernel, which we show here on the right, which we've actually fit to the data. And then you pass that through a nonlinearity and convolve with a postsynaptic response, which we've also fit to this data I've shown you. And when you do this, you get something that's really fascinating. So at 1 hertz stimulation, this is just showing presynaptic spike train, in black. Then if you look at the bottom, the GERK response shows almost no outward current, effectively. Whereas at 20 hertz, you actually see this really large conductance start to appear. So this really maps nicely onto what I just showed you. I don't have time to go through all the data, unfortunately, but we also did some classic paired pulse experiments. We show that this STP model fits this data almost perfectly. This effectively could act as you could think of this as a training set, because this is actually the data that we fit the kernel to. We also tried to use this model to explain a completely different set of experiments where we found essentially meta facilitation. So really odd dynamics where the past history of facilitation influences the future facilitation possible. And so this data was not fit to the model at all, but we show that the model can almost perfectly explain it. So we have a really nice correspondence between the model and the experiments. So next we want to look at what I've described these really odd plasticity dynamics. What does this actually do for the network function? And so to do this, we made a sort of a toy model of Raphe just receiving habenula input. So these are 5,000 serotonin neurons. A subset of these receive habenula input. And most importantly, the recurrent inhibitory connections actually have the STP model I've just described in them. So the entire model is actually put into these. And we show that this short-term plasticity can actually support nonlinear input-output transformations, which is pretty interesting. So just to give you a sort of a taste of this, at 5 hertz, habenula input shown here, you really get a sustained serotonin neuron response and almost no GERK. Whereas at 20 hertz, you actually get an early peak followed by a really profound decrease in firing rate, effectively, which goes almost to baseline. And this is because the STP dynamics start kicking in, and you get an absolutely massive inhibitory response. So this is just characterized down here. And so what you see here effectively is the late response. So this 200 to 400 millisecond segment with STP, you really get a crash in the response as a function of the habenula output, right? And if you take out all the STP rules I just told you about, but keep the recurrent network in place, all of a sudden this becomes completely linear. So it really modulates this input-output transformation. Just to tell you briefly, another really interesting thing that these recurrent inhibitory connections with STP can generate is input gating rules. So what does this mean? If you have a network where part of it receives input from habenula and another part receives input from input A, you can then, so here on the left, you see input A can generate a nice response in serotonin neurons, but if it's preceded by input from habenula, shown on the right here, then you effectively get complete removal of the response of A, and that's shown here. And if you take out STP, all of that goes away effectively, and you have no modulation of separate parts of the network effectively. So from now, up till now, I've told you that at low input frequencies, you really have direct excitation that dominates, and at high input frequencies, you have recurrent inhibition. So what is the relevance really for the serotonin system would be graded threat processing. So I'm going to very quickly go through a simple head-fixed classical conditioning task. I don't have time, I think, to do it all, but we wanted to test the frequency predictions effectively for processing and behavior. So here, simple task, the animal's head fixed, it gets a cue and it later reward, and it learns to anticipate. So here, just showing the licking, anticipatory licking, there's more for large rewards. And then what we can try to do is stimulate this pathway at different frequencies and look at the effect on modulation of anticipation. And so what we see here is that, if you stimulate this pathway at 20 hertz, you effectively get almost complete suppression of the licking response, of the anticipatory licking response. So that's shown in blue compared to black as non-stimulated trials. And at 5 hertz, you actually get absolutely nothing. So this maps really nicely onto the short-term dynamics I've just told you about, right? Where 5 hertz excitation would dominate, 20 hertz inhibition would dominate. This is just some group data here. And so the conclusion really is that there's this recurrent inhibitory network acting over behavioral timescales and really governed by these short-term dynamics. And it filters aversive information to modulate some sort of behavioral anticipation. So I just want to say thank you for everyone involved, especially my supervisor, Dr. Jean-Claude Bake. Here's other people involved. You can follow me on Twitter if you want, or ask any questions. So yeah, thank you.
Thank you, Michael. That was a really lovely demonstration of modeling and physiology. It was a beautiful combination. And
yeah,
we have people who are just really shy in the audience, but it's not going to stop me from asking or commenting. Okay. I think that this is your work shows how important it is to be testing across all the range of frequencies. And I wonder to what extent how much of this kind of behavior has been missed. But it occurred to me, I mean, you're working in Raphé and Hebelian networks, but it occurs to me that this is almost like a cellular protective mechanism. For instance, in epileptogenic tissue, you have a runaway effect where there is no inhibition. And it just occurred to me that maybe something like this might be acting elsewhere to protect people from these kind of epileptic-like phenomena. Now, what do you think about that?
I mean, yeah, that's certainly really interesting. And I guess it is true that Raphé itself has a really low firing rate of a few hertz typically. So that's why it's thought that this current was actually auto-inhibition, so the cells inhibiting one another. I think it could still play the same role you're describing at the level of the network, right? Even if the neurons aren't directly inhibiting themselves, they might inhibit their neighbors to sort of regulate overall excitability. So yeah, that's a really good point.
Yeah, you might want to try testing it with anticonvulsants, just for interest. Yeah. Okay. Well, thank you so much, Michael. That was really great. And so now we move to our last speaker, who is Vicky Zhu. And she, again, is going to be talking about nonlinear behavior. And this time we're talking about semi-balanced states in central nervous system circuits. And again, very, very interesting. And congratulations on this work that you're talking about is actually a PLOS comp biology paper. The other two papers we featured from the other two speakers address different work, but Vicky is talking about the work in this particular paper this time. So Vicky, without further ado, take it away.
Thank you very much for introducing me. Let me share my screen. Okay, hello everyone, my name is Vicky Zhu. Today I'm going to present this nonlinear computations in semi-balanced networks. This work is developed through my advisor, Dr. Rosenbaum's lab at the University of Notre Dame. The idea of approximately excitatory and inhibitory balance has been widely observed in this cortex. As you can see through down below here, many literature has demonstrated this balanced idea through their experimental cortex recording. One of the questions that our lab asks is that how does this balance shape the functionality of neural responses? So let's check out, what do I mean by balance, first of all? So what I actually mean is this total current input I here, I denote, mathematically can be decomposed into two kinds of sources. As you can see from the left diagram here, one of the sources is coming from the external input, I mark in green arrow here, and the other one is the strong recurrent input, which is the blue and the red arrow here. Since the cortical neurons receive a large number of synaptic inputs, however, the synaptic weights are not so weak, so we have moderate synaptic weights. So this means we can scale for those two resources I talked about earlier by the average strong coupling strength, which I mark in JK bar here. So the balance in this situation really means just a cancellation among those sources. As you can check out from the right plot here, we see a total current input marked in black here stay relatively small. So this balance notion give a rise of the firing rates as a one-to-one function of synaptic weight W here and stimulus input X. Okay, and this analytical expression of the rate has been widely used to study this cortical circuits responses. through many literatures. However, it has some issues. One of the problems that we face is that, as you might notice, the rate a linear function of the stimuli. So that means x in term x and r, the relation is linear. However, what do we know about our brain? It's that it are capable of performing nonlinear computations. What does it mean? It means that the classical balance cannot explain this phenomenon very well. So another problem is that the parameter space, the W, must be carefully adjusted in order to produce a non-negative rates. And this is really hard because when you have more subpopulation or cell types, this condition will be violated. And we were able to prove that for a specific network, for every W that if satisfy the Dale's law, there will be external input X will violate this condition, which means it will produce a negative rates. Let me illustrate these ideas in a simple model by consider two external population input, namely X1 and X2 here, into this spiking recurrent network. Okay, contains two excitatory populations, E1 and E2, and one inhibitory population, I here. We are going to transition in between these two different types of stimulus. Let's begin with stimulus input that has the equal firing rate, which I mark in triangle here. So that means the Rx1 equals to Rx2 here. What we see is a balanced state into the network. So we can see that for three population, we have a strong excitatory and strong inhibitory input to the network. And the black ink in the middle here is the total recurrent input for these three populations. They stay relatively small value and then concentrated through time. And as a consequences, we will see this irregular and asynchronized spiking activity in the raster plot on the lower left here. Now, let's transaction into another different stimuli type. So if I increase this Rx2 a little bit, and then what we see is access inhibition to E1 population. Okay, so as you can see that total recurrent input lean towards to a negative value here. However, interestingly, we see the remaining population, so E2 and I population forms a balanced subnetwork. As a consequences, check out the raster plot on the lower left. We see due to this access inhibition to E1, we have a silence behavior in this spiking network. And then just because of the access inhibition, neurons in that population no longer spike, so we can see it has the silence. And as corresponding to that, the other population, there is a slightly amplification for the firing rates. This is due to the offset of the spiking of the silence in E1 population. Okay, those two properties I just addressed give us to define the notion of a semi-balanced state. What do I mean by that? It's when the balance is broken, we see that access inhibition to some population. We also see the remaining populations form a balanced subnetwork, and we call this a semi-balanced state. Now, our lab can further ask the questions as such. The semi-balance, how does this semi-balance shape the functional properties of neural responses then? If we step back a little and redrive the rate expression under this new assumption as this strong coupling strength limit, what we see is this implicit expression of the rate, of the firing rate, in terms of a rectified linear. And this is interesting because this expression, it's also frequently used as activation in artificial neural networks. And also notice that there is a linear mapping in between stimuli, which is X here, to the rates R here. And more interestingly, this is can be directly related to the fixed point firing rates in the ReLU recurrent neural networks, and has been studied by the Kriena-Kurtel group for this threshold linear networks. Although this exact expression of the firing rate is in this strong coupling string limit, it does give a good approximation for biological realistic networks. And you can check out some nice work from this from other literature. If we return to the examples with multiple populations, which I talked earlier, I want to examine this, I want to test the efficacy of the theory. So we can see the simulation here as a solid line matches pretty well with our theory. But more interesting than testing the theory, if I take a linear readout of this system, it's easily shaped into this continuous XOR function. Okay, and then one interesting about this XOR function, it's one of the simplest nonlinear type, and it's cannot be linearly separable. And so far, what I have talked about is on the level of average across neuron populations. If we extend this balance notion into a more detailed level, namely this cell level, by looking at the individual neurons' total current input. So what do we see? It's spread out, like for the 10 seconds for the first stage here, we see a huge spread out distribution of the synaptic current. As a consequence, we see a flat tail distribution for the rate. So this means in the detail level, we see an imbalance. But luckily, the work at Vogel's group in 2011 can save us in this situation. They introduced this homeostatic inhibitory spike time dependent plasticity rule, which can retain the detail balance level. So upon a few seconds activation, what you see is that the total current input converge to something very concentrated value here. So the black curve in the middle, you can see it's hovering around something very concentrate. What I have talked so far is by consider this time constant input, but in a more realistic biological concept, if we consider this time-dependent stimulus, we can see all the properties I talked about, the semi-balance will be achieved in this detail level as well. So as you can check out in stage 3 here, the total current input has when we break the balance, it will lean towards to some negative value, and then the remaining sectors, some of the sectors form a balanced subnetwork. So all this is together telling us that together with this plasticity rule and introduce the time-dependent stimulus, what we have is this detailed semi-balanced state. We further investigate in the 2D stimuli into ND firing rate by plotting this joint distribution of firing rates together as with this parameter stimuli vary. What we see here is this rich and nonlinear stimuli representation neural manifolds here. And this nonlinear stimuli representation can be precisely also quantified by this dimension reduction technique by check out the PCA and isomap. PCA is to find this linear manifold namely this hyperplane. However, this isomap is to use this nonlinear manifold. So as you can see, the plot below here, we see that more dimension is needed for PCA compared to for isomap only takes 2 dimensions. So what is saying is that the firing rates really lives in these 2D nonlinear manifolds. To wrap up of what I have to say on this talk, I first introduced this balance network. It's linear, and then we see it has to satisfy this non-negative firing rate. And due to those constraints, we were able to extend it into the semi-balanced network, and then giving an implicit expression for this firing rate in terms of rectified linear. And notice it's a nonlinear mapping between stimuli to the firing rate. In the individual cell level, we see an imbalance for the network, and the homeostatic inhibitory spike time dependent plasticity can retain the balance to the detail level. Together with the plasticity rule, we introduce this time-dependent stimuli, and it's naturally we retain this detailed semi-balance. So the take-home message for today is that in the semi-balanced network, nonlinear stimuli, we will see this nonlinear stimuli representation. If you want to investigate further for the work, you can check out this paper we recently published in PLOS, Computational Biology, and this work is collaborated with a graduate student myself and the recent graduated fellow Dr. Cody Baker and my professor Dr. Rosenbaum, and funded by various resources including NSF and NeuroNext. I'm ready to take any questions.
Thank you, Vicky. We do have a question from the audience, and it's a question from Mega Gosh, and he asks, can you explain how a semi-balanced state differs from sparse firing network?
Okay, the semi-balanced state is really in the notion, let me go back a little bit. Yeah, the semi-balanced state here is in the notion that extend this balanced network when those conditions that fails. For example, we have for the balanced state that we see this linear expression in terms of stimulus and the rates. And we also have to make sure that the firing rates has to be non-negative. And so we introduced to this semi-balanced state by when this balance condition break. So means that in the axis inhibition level, when balance broken, which means this firing rate, so this expression, firing rate has been broken, we have a negative firing rates, and we will have the semi-balanced state. And another interesting condition for the semi-balanced state is this subpopulation from like a balanced subnetwork. So excluding that population has axis inhibition, the remaining population will still form a sub network. So what your question asking about the difference between this semi and the sparse. So this this toy example here, okay, is to examine some how to say this, like we can subdivide it into different if this expression really break, this rate become negative, it really happens when you have a subpopulation, more subpopulation or more different cell type. So this will break. That's why in this toy example, what I examine is we have, I have this E1 and E2 and I a more sub cell type. So that's how we come with a semi-balanced network.
Okay, well, Go ahead. Sorry, Vicky, finish.
Do you have any?
So that, yeah.
I was reading that.
Yeah, so we are out of time, but thank you very, very much to both you and Michael and Vicky for some really fascinating presentations on nonlinearities in the nervous system and how we can simulate those behaviors. And I also would like to thank very much Aditya Iyer, who did an absolutely spectacular job on the hosting side and doing the back end side of this session. So I hope you enjoyed it. Hope to see you at another session. And so enjoy the rest of the week. Thank you.
Thank you.
Thank you.
I was not thinking properly, and I didn't realize you also had to scroll to the left and right for the calendar. So I missed like a few talks that were on the far side of the calendar, and then I was like, oh no. So yeah, I'm gonna be definitely gonna be on the YouTube channel afterwards.
All right, shall we get started? Okay. Welcome everyone. My name is Shankar Ragappan, and along with me I have Elena Jones. We're running this session today. Now we're excited to learn from our colleagues how the brain learns using different task-relevant information in this session. And we have an interactive session, which means the speakers spend about 5 minutes introducing their topic, following which we have about 10 minutes for Q&A. Since we have only 2 speakers today, we can try bringing attendees on stage to questions. So please mention that you would like to ask questions on stage by typing on screen after your question. And if you're camera shy, you can just use the Q&A panel, and don't forget to vote for your favorite questions. Having said that, I'll keep the introductions brief so that we have plenty of time for the talk and Q&A. First up, we have Jennifer Ruttle from the lab of Denise Henriques at York University, and she'll be talking about motor learning. Take it away, Jennifer.
All right, let's get my screen shared with you all. Okay. Let's make this up. All right, hopefully everything's good to go. All right, okay, so as we said earlier, my name is Jennifer, and today I'm going to be talking to you about hand localization shifts after different types of visual error feedback in reach training. So just to like get you guys all on the same page as me, everyone has hopefully some of the same knowledge. I'm just going to do a little bit of background on a model I'm going to be talking about. Specifically, this model was proposed by Maurice Smith in 2006, and it's just a simple 2-rate model that says or proposes that adaptation is a combination of 2 separate processes, okay? So the first being a fast process, which is this solid black line in the figure below, which learns fast but also forgets really fast. And then there's also a slow process, which is the black dashed line in the figure below, which learns slow but also forgets really slow. And recently, these 2 processes have actually been attributed to explicit and implicit components of learning. And realistically, and what it really is, it's like this balance between this fast and slow process, or this explicit and implicit components that create the learning or actual motor behavior, motor output that we see people making. And that's what that purple line is on the graph on the left below. And so I'm just going to be generally using this model to talk about how, or to kind of frame how learning may be different when there's different visual feedback cues during the adaptation phases. So my participants did the exact same paradigm as what we just saw. They started out with an align phase where the cursor perfectly matched the hand. We're going to be doing a visual motor rotation here. So the hand and the cursor were perfectly matched for the align phase. Then we introduced abruptly a 30-degree rotation in one direction for 160 trials, after which we shifted it or flipped it in the opposite direction. And then we, on this dashed line, we're back at what we call clamp trials. And so here, the cursor always went directly to the target, regardless of where the person moved. This ensured that they were not getting any visual feedback, and thus there was no visual error during those trials. They were just moving straight, the cursor moved straight to the target. And so, as I said, we wanted to look at how visual feedback might change the model. So we had 3 different kind of visual feedback groups. So the first one's the common one that we all probably know, and this is what we called continuous feedback. So the cursor was visible the entire time the participant moved out to the target. And if we see on the left here, they did great during aligned, and they were able to learn the rotation really fast. They tried to learn the counter rotation, and then they show us this really nice and iconic kind of rebound during those clamp trials. Rebounding back to this performance they did during the first learning phase based on the slow process of the model. And then our second group is what we call the terminal group. The cursor was not visible the entire time they moved out to the target, and it was only visible once they stopped at the actual target. And so we can see here that their movements were actually very similar to the continuous group. So even though they weren't getting that visual feedback during training, they were still showing pretty similar trajectories, which was great. And then our final group's a little different. You might notice that there aren't any reaches for the first 3 phases, only during the clamp phase. And that's because this group was actually not allowed to choose where they moved. They were constrained in their pathway, and thus they were only exposed to the rotation because the cursor always went perfectly to the target because they were perfectly countering the rotation. And this means that they didn't make any, they made no errors during these first 3 phases, but during the clamp phase, we did let them move volitionally, and we can see they're producing pretty much identical rebounds or reach trajectories as the other 2 groups, indicating that even though they didn't get to move around during those first 3 training phases, or we're still seeing similar shifts. And so from this, we're saying, okay, well, even if we just look at the actual data itself, it looks like the 3 different types of visual feedback didn't really change training very much. And so we actually, for the first 2 groups that we had training throughout the entire way, where we had reaches the entire time, we fit the 2-rate model to them. And here I've just uploaded the model output, and we can see both lines are basically right on top of each other, indicating that not only are the reaches and the actual behavior we're seeing very similar, but even the model output is very similar between these 3 different feedback conditions, or the 2 conditions. So getting to those hand localizations. So after every training trial, we also ask the participants, where do you think your hand is? And so once again, we'll have the same kind of paradigm here. And what we did was basically, after a training trial, the right hand, which was hidden from view and holding the robot, was dragged out to a location on this arc that was visible in front of the participants. The arc was as far away as any of the reach training targets were, so they were just dragged out to a location out in front of them with their right hand, which was hidden from view, and then they used their left hand to indicate where they thought their right hand was. And so if they thought their hand was exactly where their hand actually was, we would see a straight line across 0, indicating that they were right on target and they were always perfectly right. But when I layer the 3 groups here, we can see that they're not flat. And in fact, after really only 1 rotated training trial, we're seeing this pretty big significant shift in where they think their hand is. And not only that, but it also shifts when they get that counter rotation. So this kind of made us think so they're just gonna shift where they see their hand is the second they get this different rotation, right? But what can we do to maybe break that? How does this really happen every time the rotations shifted or changed? And what about if it's different sizes and different directions? So for our next kind of experiment, what we did was just that. Every 12 trials, we changed the magnitude and the direction of the rotation. So sometimes it went more negative, sometimes it became positive and changed directions. But this way we could see how well they could learn with the constantly changing rotation and where did they think their hand was. And so here we can see they actually learned really well. Every time the rotation changed, they tried to learn it. They put in that effort and really went for the new rotation. And they even shifted where they thought their hand was. It was incredibly fast and awesome and surprising to us to some degree, which means it just wants to keep shifting, and who knows how we can stop it. But generally, yeah, it's from all of this, we're kind of seeing that these shifts in where you think your hand are synonymous with motor learning, regardless of the cursor feedback that you experience during training. And that's it. Thank you.
So that's the applause on behalf of the attendees. So questions. People please feel free to ask questions. And if any of you want to come online or on screen, do mention it in the chat. In the meantime, so when you, let me think through this question properly. So what you're showing is that there is an internal way that people can identify where their hand is moved to, right? Like a proprioceptive.
Exactly. Proprioceptive, you know, like the muscle spindles about where your arm is in space, the fact that you're holding the robot, as well as there's a prediction about you. So the main thing is that since we're not letting people actually choose where they go, they weren't able to make any sort of like efference copy or idea about where they were going to move. So when we're measuring where they think their hand is, we really think we're measuring as pure as we can get to just what that proprioception is saying, because we're not letting them choose where they go. Their hand is just dragged out and then they have to indicate that.
Okay, got it. So we have some questions here from Jaran. Do you want to come online? Or if not, I can just, I'll just go ahead. Okay, so how well does the model predict the fast changing target?
So if I'm understanding this correctly, right now we actually don't have, there's nothing in the model that we don't tell it anything about target. We in fact actually, we have 4 training targets they reach to. They're all within 60 degrees, but they're not, we don't look at them differently. We just combine them all. So at this point, there isn't any like kind of consideration for target, but I do know that the original McDougall paper where they attributed these to explicit and implicit components, they did either having the targets in like one quadrant or spread around the whole thing. And the model did show slightly, did show a difference in the balance between the fast and slow process when you add more targets. We have also, like, one other thing that would be interesting is actually just like, because we're not having them, the original model was assuming that they were only doing reach training trials. Now we're having them do a reach training trial, then a localization trial. So this is different. And at this point, I still haven't figured out the best way to change the model to like account for that like time lag or something like that. But that is, I hope that answered your question.
So next we have Nick, and Nick would like to come online. So Lena, can you help him come online?
Hello, can anyone hear me? Hello, yes. Hello. All right, thanks for the talk. That was really interesting. My question was just about the exposure group. So were they actually making any movements, or were they just visually observing these like fixed trajectories that you were talking about?
Yeah, great question. And there is some papers that like have people physically just watch it and do what they call like observing, but we actually did have them moving. So they, because something I didn't explain is that there was a time constraint. We wanted them to move pretty quickly. So they had to like reach the target within 800 milliseconds. So we didn't let them move left or right, but they just had to like move as far away as they could. And basically there was a pillow at the end stopping how far they could move, and almost like a pillow on either side. So they got to move, but they really had no choice over where they moved or even realistically how far they could move. They just had to, because they had to go to the end of the target. So they didn't get to move, but we have done, in my previous experiments, I've done where they didn't move at all and we just dragged their hand and we had them move and drag, and we've done all these different combinations and there isn't much difference. But this time they did some movement, but they didn't choose the direction.
Yeah, so sorry, so you see a relatively similar amount of proprioceptive recalibration across all those different variations that you were just describing?
Yeah, very similar across them. Like it seems, because the main thing we think that drives this change in proprioceptive estimates of hand location is really this discrepancy between you see the cursor in one location and you feel your hand on the robot somewhere else. And there's that visual proprioceptive discrepancy that like is kind of what we think might be driving some of this to some degree.
Cool, great, thank you.
Great question, thanks.
All right, so next we have a question from JJ Orban. What is the link between the two phenomena, shift in hand and adaptation? Does the fact that it happens in one trial suggest that those are completely independent? Because adaptation does not really happen in one trial, even though there is some one trial adaptation. And unfortunately, it cannot come online.
So this is a great question and is honestly something that we are really trying to like dive into, because you're right. At first we were for a while before we had this like, and we realized it was so immediate, we were thinking that maybe hand localization shifts something, because to some degree, I would imagine that when you're doing this adaptation, I can see that if you shift where you believe your hand to be, that may affect where you think your start positions are and all of these things. So that's why we do think that they're definitely mostly independent, I would imagine, but there's got to be some bleed over of like, because we see them happening in the same context. You're right, there could be a third variable that we're not even capturing, but it does seem like there seems to be something that this change in where you think your hand is affects adaptation to some degree, but we're just not sure, which is why we really want to see if there's ways that we can stop the shifts in felt hand position from happening. And if adaptation still happens, and slow it down and things like that, but we haven't seemed to be able to do that. So maybe that is kind of proof that it happens really quick and it changes your start position, which then has some effect on adaptation throughout more training, but something we definitely need to look into more. Thank you for the question and coming on this late if your family's asleep.
Great. We have a question. We have a couple more questions. One from Jonathan Say, which has, I think they're split between the chat and the question and answers. Do you think changes in hand localization drives motor adaptation? That's the first question. And is there a reason why hand localization saturate around plus or minus 5 degrees?
So I think yeah, like we definitely do think that this change in felt hand position is driving like adaptation to some degree. We do think that it goes in that direction as opposed to adaptation to shifts in hand position. We think that the arrow would go the other way. This whole capping thing that you're suggesting is so interesting to us, because we have like questions about like, if we do a stepwise paradigm, can we increase this? But I think to some degree, it naturally makes sense that if my hand is here, I'm not going to believe it's like 7 miles over there. There's got to be some physical limit to that. So it does seem to be limited to about 8 or like, yeah, between like 5 and 10 degrees. It seems to be that. We aren't entirely sure why, but it may just be proprioception being like, I can't believe my hand is that far away.
Great. One last question. So do you think the proprioceptive estimate task could be used as a measure of explicit?
You know, it would be nice if it could, because it's so pure, but I've been in this lab for a few years doing this task for a while, and I'm doing it, I'm like, I know where my thumb is, I'm perfectly, and I always am off. So this is implicit, because even me being like, I know what I'm doing, you can't fool me, I still show the same shift. So it's definitely something going on implicit that we don't have access to explicitly. But yeah, really good question.
Great. So if you have more questions, we can always ask after the next talk, since we have about 15 minutes left. So I'm again an applause for Jennifer for her wonderful talk.
Thank you.
Next up, we have Michael Stein from the lab of Carl Olson at the University of Pittsburgh. Take it away, Michael.
Great. So yeah, my name is Michael, and I work in the lab of Carl Olson. And today I'm going to be talking about surprise signals in the macaque frontal eye field. So in order to begin, we're going to just discuss prediction error very quickly. Prediction error is a neural response that signals that an expectation has been violated. And there are two influential models of prediction error. One is the reward model, in which the sign value of an unexpected outcome determines the neural response. And this is basically the classical reward prediction error, if you're familiar with the Schultz literature. And just as a toy example here, I'm going to just kind of illustrate what these kind of models predict. So say your boss gives you a manila envelope, and usually when you receive a manila envelope, it contains documents that you have to sign, then you submit to the accountant, but it's sealed. When you unseal the manila, and so basically your expectation is neutral, whether it's not good or bad. And here you have the neural response, the expectation is neutral. When you open it up, and maybe there's a bunch of cash in there, that's a great surprise for you because your boss says you can keep it, this cash, and you weren't expecting it. And the reward model would predict neural response enhancement in this case. However, say you're back at the manila envelope case, you have a sealed manila envelope, and your expectation is neutral again for what's going to be inside of it. Once you open it up, if you get a snake inside there, that's a pretty bad surprise because that puts your life in danger, or your boss is pulling a prank on you. And the reward model would predict that you would get a neural response suppression that scales with the value of the surprise. The competing model I'm going to call the surprise model or the attentional capture model, which is that in the unsigned magnitude of the attentional capture might determine your response. And in this case, if you get a good surprise like a bunch of cash, you'll still see an enhancement of your the neural response. But the critical difference is that if it's a bad response, you might see also a neural enhancement because, again, the surprise captures your attention in either case. Whether it's good or bad, it captures your attention. So these are just two different ways that the effective surprise might manifest itself in neural activity. So we were interested in how neurons in the frontal eye fields are respond to surprising outcomes, because neurons in the frontal eye fields carry both attentional and reward-related signals, but their response to surprising outcomes is unknown. And these are visually responsive neurons in the prefrontal cortex of the macaque monkey. So in order to do that, we developed a simple reward surprise task in which the animal is passively fixating, and we shine visual cues into their response field. It's a probabilistic task in which the animal is fixating, and two cues in succession are presented, in which 83% of the time, if you get a good leading image, you're going to get a good outcome. You're going to get a big reward, okay? And again, there's no behavior here. However, because it's a probabilistic task, 17% of trials, you're actually going to get a bad trailing image, which signals with 100% probability they're going to get a bad reward. So similarly, we have bad leading images, which usually signal that you're going to get a small reward for the animal, but sometimes, rarely, signal that you might get a good surprise if they're followed by a good trailing image. So what we find here in the neural response, since we present, again, we're presenting two images in succession, we're interested mostly in the second image, but just as a sanity check, we can see that the leading image, when it's likely good as opposed to likely bad, we see an elevation in the neural response. This has already been described in the frontal eye fields. But what we're really interested in is the response to the second image, and whether it's expected good or surprising good. And when it's surprisingly good, we can see a big enhancement in the response. Similarly, surprising bad cues turn out to also enhance the neural response. So this seems consistent with our attentional model, in which both surprising good and surprising bad cues enhance the neural response, and it's not a signed effect that we see. And we find that this surprise effect is consistent across neurons. Here I have two epochs shaded because we're interested in the dynamics, which we might get to if we have time. But basically, it's a pretty robust effect across neural cells. And in the good surprise case, you see it early and you see it in the late visual response. However, in the case of bad surprise, you see an enhancement, but as is obvious, should be obvious here, the dynamics are different. And basically you see a big enhancement in the sustained response and not in the early response. And so in this case, I decided to take a quick look at the pairwise correlations in the sustained response. And since this is the one that seems well characterized or easy to interpret. And we find that in the sustained response, we see a reduction in noise in pairwise correlations between neurons so basically surprise reduces pairwise correlations. In the visual response, however, we find something a little more possibly interesting, which is that we see evidence of a signed prediction error in the early visual response, which is not present at the level of the PSTH. And like we saw earlier, we did not see that emerge in the bad surprise case. So it seems that there are signatures of signed reward prediction error in the early visual pairwise correlations. So the take-homes are that surprising cues enhance the FEF neuronal response regardless of the valence of the outcome. Enhancement is consistent, feature shared among most visually responsive neurons in the frontal eye fields. Surprise reduces pairwise correlations in the sustained visual response, akin to other demonstrations in the case of attention. And taken together, these observations are consistent with the idea that neural activity in the FEF reflects the capture of attention by surprising events. So sorry, that's all I have.
That's good. That's the applause from the attendees. So as we are waiting for questions, I had a quick question about one of the plots. So the one where you showed the response to the surprise for the good and the bad, it seemed that the good surprise had an early response, whereas the bad surprise seemed to have a later. Is that?
Yes.
Am I seeing that right?
Yes. Can you still see my screen here?
Yes.
Okay.
Yeah. So this one. Yeah. So yeah, the bad surprise, the increase seemed to happen a little later. Is that right?
That's correct. Yeah. So the dynamics are different, right? In the good surprise case, you have a big increase early. In the bad surprise, you have pretty much no effect in the early period. So what we could be seeing is potentially two different phenomenon. And that's why I decided to take a quick look at, or I decided to explore the correlational structure. And we found that in the pairwise correlation activity, there is an element, of that surprise.
Got it. So, like, do you know why this happens? Like, do you have any ideas of why the correlations are different?
It's a great question. So the work on pairwise correlations is really very new. Just something I've been working on very recently. So I don't have a good interpretation for why the information in the pairwise correlations are different than information in single neurons. But a number of studies have suggested that the information in pairwise correlations could actually have a closer link to behavior than just the neural responses themselves.
Yeah, that's interesting, right? So I mean, it's not like a reduced or increase. It's just that the direction seems to have flipped.
That's right.
Yeah, that's really interesting. Are we looking for questions? So, like, what do you think like the next steps are in this work?
Yeah, it's a good question. So, I mean, I'm really interested in examining the dynamics, like you said, because there is this clear difference between the good surprise and the bad surprise data, and really just trying to build a model that captures why we might see this difference.
Do you have, I mean, I'm not familiar with the literature in this area, but are there like any interesting like avenues that look promising or that look interesting for you?
Yeah. Yeah, it's a good question. So there's been some work done actually locally here about projecting this data into kind of into state space and seeing whether the a big question I have, and it's an analysis I'm still working on that I'm not finished, is to determine whether the value dimension and the surprise dimension are orthogonal in state space. And if they are, basically, you could assume that a downstream structure is able to basically read this signal regardless regardless of the single neuron responses. That it might be a population signal. So that's kind of, yeah.
Cool, nice. Yeah, so I think that's about it. Do we have any questions for either of the panelists right now? If not, thank you for joining us, and another applause for the panelists again for the wonderful talks. And I think that's about it. So we'll see you sometime later.